I am very excited to chat with today's guest. We have Dr. Sam Shay. We are going to be talking about genetics and methylation. Going to be a fantastic conversation. So let me go ahead and dive into Dr. Sam's bio here. So Dr. Sam Shay solves health puzzles for busy, health-conscious moms, mompreneurs, and adults with Aspergers so that they can exit survival mode and re-enter community. Known as the friendly lab nerd, Dr. Shay specializes in functional lab and genetics analysis for data-driven results. And Dr. Shay is also a stand-up comic using clean observational comedy to educate and entertain. And thank you so much for joining us, Dr. Shay.
Yeah, thank you, Dr. Zemsky. I'm really happy to be here. It's fun to do these things live, and my hope here is to clear up a lot of confusion around the concept of methylation. We'll also touch on, I know your specialty is thyroid. I can bring in, I have whole separate, I've been on multiple Hashimoto summits and have a big interest and deep dive into various parts of thyroid health and Hashimoto's, but we'll focus on methylation. The reason why is that there was a podcast late last year where Gary Brecka went on to Joe Rogan and talked about methylation, and he spoke about like these 5 methylation genes are all you need. And I just wanted to create a bit more of a rounded picture on the concept of methylation, because I do respect Mr. Brecka's ability to take complex information and simplify it, make it very teachable and understandable. The overall criticism is that he vastly oversimplified and frankly ignored some higher level genetic phenomenon that is more important than methylation. And there are things that are more important than methylation. And the way we know things are more important than methylation is because the body will pivot and shift methylation processes, because these other things are more important that they have to address. And I brought with me, because the concept is, because methylation's a complex space, and there's a lot of complexity in the fields and in the people talking about it, I brought with me a presentation to show my work, to show my homework on why I disagree on certain really meaningful parts of Mr. Brecka's work. And again, he's a great educator. I don't ever take that away from him. He's got a very fascinating background as kind of like a medical actuarial, like he worked for the insurance company. It's kind of this weird hybrid between an insurance adjuster and actuarial person. He's a very interesting perspective. I respect his perspective. He vastly oversimplified, and I wanted to kind of, in my opinion, correct the record.
All right. No, awesome. Yeah, you hear a lot about methylation, but as you mentioned, it's not the most important pathway. There's but why? I don't want to say it's like hyped, but why is it like why are so many practitioners, including myself, talking about methylation compared to other things that you'll bring up?
Well, methylation is extremely important. I mean, what is for people who don't know, methylation is referring to a methyl group. A methyl group is a single carbon with 3 hydrogens attached to it. And it's like the simplest addition you can make to a molecule next to like adding just a straight up hydrogen. And what methylation is, it's a complicated form of tagging to molecules to mark them for starting or stopping certain things. And the reason why, when you add just 1 carbon atom with 3 other hydrogen atoms attached to it, you think, well, how is that possibly that important? Is because structure dictates function in the human body. And when you alter the structure of a molecule, even by just 1 carbon with 3 other hydrogens linked to it, you alter its function. And so methylation is critical for signaling to other systems of the body. This molecule is earmarked now for this purpose, or is now quarantined away for the purpose it originally was for. So it's very, very important. And the problem is that there's a primacy of, it was kind of one of the first it's kind of like the founder effect, that MTHFR was one of the first most famous genes that got into the public consciousness when genetic testing became more available to the wider public. And because MTHFR gene just rocketed up as one of the most significant early access genes available to test, it just became this fixture in the not only scientific community but in the public consciousness. So many other genes, in my opinion, are much more important than MTHFR, but that founder's effect has lasted. And I think we're just dealing with this genetic inertia of the founder effect of the MTHFR gene. That's my opinion, and I'd be happy to see evidence to dissuade me otherwise. But that's what I think happened.
Yeah, no, I agree. A lot of people, including patients, just know about MTHFR. You know, they bring up MTHFR, and even when it comes to methylation, I think you'd agree that MTHFR is a component of it, but there's a lot
Oh, we're
more
going to
when it comes
see
to
a giant
methylation.
schema of about 12.5 genes. The MTHFR is going to be way off in the corner. So yeah, I agree with you.
Yeah,
Definitely.
and what? So, can you briefly talk about as far as like how many genes are out there, so people could get an idea, and then out of those genes, like how many on average do you normally test for?
Sure. So there's approximately 25,000 genes in the human genome. I mean, I've seen numbers range from 25,000 to 30,000, but the point is that there's a lot. And the criteria for the criteria for understanding what are the best genes to test for, um, I, I come at it from four specific prioritized filters. Number one, the genes I'm testing as it relates to health and well-being, I am looking for not the disease genes. I'm looking for the drivers of disease. So I'm not looking for the cancer gene, the heart disease gene, the stroke gene, the Alzheimer's gene, the dementia gene, the diabetes gene. I'm not looking for those. I'm looking for the genes that control the drivers of all of those genes. So there's 7 major drivers that when activated will poke down on these quote unquote disease genes downstream. And those 7 drivers are: number 1, inflammation; number 2, your ability to quench free radicals in the mitochondria, where 95% of all free radicals are generated; number 3 is your ability to detox properly, phase 1, phase 2 liver; number 4 is vitamin D receptors, your ability to use vitamin D, not synthesize vitamin D from the sun, which is important, but to actually get the vitamin D once it's in the blood, whether it's from sunlight, supplements, or food, and get those vitamin D molecules into the cells. And then methylation is underneath that. Like, those 4 processes are superordinate above methylation. And then there's cardiovascular circulation, and then there's fat and energy metabolism. So those are the 7 drivers. So I'm looking for genes that are controlling those 7 drivers of disease. And more importantly, I'm looking for the ones that are looking at the genes that are upstream. So what are the ones that, of those 7 drivers, there's like hundreds of genes that relate to inflammation, and directly or indirectly, but there's like 15 that are the upstream, the generals that control all the foot soldiers underneath. And I want to look at the top 15 controlling genes, not the hundreds of genes that are kind of diffuse across the whole inflammatory pathway. And that applies to free radical damage, liver detox. There's all these different genes we talked about. What are the most important genes within those 7 drivers? And the third criteria is, of the remaining genes, which genes show up in at least 10% variation in the general population. So I don't want to test for super obscure genes that only 0.001% of the population have. Now, for those people that have them, those are very important. But I'm talking for the purposes of general health and well-being, what is a type of genetic test that I believe everyone should have? I want to look at genes that minimum 10% of the general population may have variants on it. So there's a really good chance that if there's something awry, then we can catch it and not have to go down these super obscure genetic rabbit holes. And then the fourth criteria is, once you have figured out the genes that fit the 7 drivers, you find the genes that are upstream in those genetic drivers. The upstream genes in those genetic drivers have at least 10% variation in the population. Which of those genes remaining have peer-reviewed scientific research in top-tier journals done on humans, not wombats or nematodes, that lifestyle, diet, nutrition changes alone will shift the epigenetic expression in the direction we want. So those are the 4 criteria. And when you boil it down, there's like 100 or so. It's like 100-ish plus. Not a couple thousand, not a couple hundred, not even 200. And certainly not just 5 that Gary was pushing. There's about 100. And those are the genes that I look at. And again, not to the exclusion of other niche genetic tests. And for those of you that want to find out genealogical data, like what percentage Irish you are, or whatever, like that's fine, but that's not the testing that I'm interested in. It's what are these genes that are the highest priority as it relates to all facets of health. And so I test for, at the moment, I test for 110. And which is one reason why I was very uncomfortable with Gary's assertion all you need are these 5. And those 5 that he mentioned, one of the 5 I think is not worth testing, because it doesn't fall into that 10% variation thing. And in fact, the AHC3, the AHC is a Y or 3 AHCY gene is one of the most conserved genes across the mammalian, the mammalian project we have out there. And even the answer to that is B3. Like literally the only thing you can do is B3. So, which it should be in anyone's multivitamin anyway. So it seems like a really silly thing to test to begin with, because there's hardly any variation, and the answer should be a part of your stack, regardless whether you tested for it or not. So, I even have quibbles with the 5 he chose. So, did I answer your question, Dr. Osetsky?
Yeah, no, you answered the question. And so pretty much you're looking, like you said, for drivers and not genes that are determining if you're at risk for cancer or heart disease. Or again, for those, listening to this, who want to know if they have a genetic predisposition for Graves or Hashimoto's, or, I mean, most people listening to this already know, but you're not really looking to measure genes to tell a person if they have a disease, just really the drivers of disease of certain conditions.
Definitely.
Yeah. And do you do genetic testing on everybody? I mean, it sounds like you do.
I do genetic testing on almost everyone. Some people don't want to do it because they have a they get squirrely about having their genetic data even tested. Other people feel that the value of genetic testing is both short-term and long-term, and it's the highest value test of any test out there, I think, in existence, because the value of the data doesn't decay over time. You can run a genetics test once, and those results stay the rest of your life, unlike, say, a thyroid test, which you have to get retested, whether it's several times a year or at least once a year. So you have to keep retesting. Genetics, you spend $1,000 on a genetic test, and you amortize that over 20 years. It's like $50 a year. So it's over time. It just the cost goes to negligible. It's just negligible. Whereas the functional tests, you have to keep repeating. Now, the value of doing functional tests in tandem with genetics tests is that what genetics tests will tell you is where's your ideal, where's your ideal diet, what are the hardwired food triggers you've got, what are the hardwired issues you have with inflammation, free radical damage, vitamin D receptors, all those things I mentioned. And then what are the genetic fault lines you've inherited that you can shore up? And so it's giving you where's your ideal and where's the things you got to shore up. But what genetics doesn't do is it doesn't give you the thumbprint of what's your biochemistry doing now. It doesn't tell you what amino acids you're deficient in. It doesn't tell you what your thyroid levels are. It doesn't tell you, what infections you got in your gut. It doesn't tell you, what your cortisol levels are over the course of a day. It doesn't tell you what, your sex hormone binding globulin is. It doesn't tell you what your blood sugar is. Like all those other functional tests are amazing and great. Like doesn't tell you what your mitochondria function, if you're doing like an ion panel from Genova or Nutristat, if you're overseas or whatever it might be. The genetics test is not the thumbprint. It's the filter in which all your physiology is translated through. So they go together. And people who choose to delay genetics testing, they've got some really acute issue going on that they have to just allocate their resources in the moment towards what's going on functionally, what's going on in my gut right now, what's going on with my thyroid right now, what's going on with my adrenals right now, what's going on with mitochondria right now. And I know genetics is super important, but I have to prioritize resources to the thumbprint of what's going on with physiology right now, because I'm in a real or perceived crisis that I need to get figured out, and we'll do the genetics once things have calmed down. So those are the cases where I don't run genetics on everyone right away.
Okay. So most people you do, some people you don't. And then it sounds like either way, you integrate the functional medicine testing. So it's not like your patients just get genetic testing and that's it. You'll also recommend other tests as well.
The thing is like genetics is one of those things where people can come in and get their genetics regardless of their age, their state of health, or their health objectives. And so it's the one thing that I'm quite comfortable, people who want to come in and get their genetics test and they have the info and they're on their way. That works for me in the sense that the data and the recommendations don't necessarily change if we're just purely looking at the recommendations on the genes alone, regardless of their health state. And what happens is that people can go and try to implement the genetics, ideally do it with support. Like the way that when I do it is I have like a 6-month program where people have support implementing, and there's a back-end course, there's an option for office hours. Like there's a way to support people through integrating all their genetics recommendations. And if people run into roadblocks, but they only wanted to do genetics, then if they run into roadblocks and the genetics recommendations hit a plateau of where they want to go, that's the time they have to examine, all right, do I really need to take a closer look at my other lifestyle recommendations that aren't tethered to the genetics, or I have to get some deeper functional medicine testing, see if there's something going on that's blocking my ability to help me benefit from these genetics recommendations. I'll give you two real examples. One is one of the genetics tests I run is looking at, are you keto, paleo, Mediterranean, or high carb? And this is not a test you can get off 23andMe or Ancestry, because it's not looking at a variant analysis. It's looking at a copy variant analysis. It's looking at the number of duplicates of a gene, not the version of the gene. And so there's a particular gene that looks at your ability to generate the enzyme that breaks down carbohydrates. And it's the duplicates that matter, not the variation. Because when you run the instruction to, like, we need to make these enzymes to break down carbohydrates, that enzyme runs through all the copies. So if I've got one, so imagine like my digestive system is a fort, and there are Visigoths, barbarians charging the gate. These are carbs. And I've got cannons lining the fort to keep them away. And the cannons are these genes that are spitting out these enzymes that break down carbohydrates. Now, the red dot, or the bad version of the cannon, shoots like 90 bullets a minute, where the yellow dot is like 95 and the green's 100. So in this particular case, the version of the cannon doesn't really matter, but there's a big difference between you have one cannon and 10. So if I have a red dot, or 90 bullets per minute or whatever cannon, times 10, that's 900 bullets, versus one green dot, that's 100. So I would rather have the 900 than the 100. So it's the number of duplicates that matter. So the lower number of copies you have of this gene, the more you are trending towards a paleo or a keto lifestyle. The higher number, it's more of a higher Mediterranean, higher carb lifestyle. So I had a perfect Mediterranean diet. I mean, I knew the names of my farmers and their chickens, and I was still having really problems with my digestion and energy crashes. And it was it just wasn't making any sense. And then when I ran my genetics, I realized I had the second lowest carb tolerance you could have. I had 2 cannons. That's it. So I'm more of a keto straddling paleo diet. And when I shifted my diet, my digestive problems of 20 years went away because it wasn't a carb quality issue, it was a carb quantity issue. And if I know my genetic tolerance for carbohydrates, then I can radically shift my diet to what I'm supposed to have from a carb tolerance level. So if I know all of these things about me genetically, I know my ideal. Now, here's tying it back to the question of if things plateau, if you know your ideal, but it's not working out. If I'm a very low carb diet person genetically, but I have to have a higher fat, higher protein diet, but I have a problem with my gallbladder or my pancreatic lipases or my ability to absorb fats, across the intestines, or a problem with my carnitine shuttle, or a problem with my mitochondria to burn fat, then I genetically I prefer fat, but metabolically I can't process it. So now we have a conflict. So these are the people that say no matter what diet I try, it doesn't work. So they can do higher fat genetically, but they can't do higher fat metabolically. On the flip side, some people are high carbs. Well, they need higher carbs genetically, but what if they have a candida infection and it causes all the candida or other infections in the gut to bloom? So they have a conflict where they need high carbs genetically, but they can't process higher carbs metabolically. So these are the people who say, I've tried every diet, it doesn't work, because they're in conflict no matter where they go. So for these people, they need to have one strategy. They need to deal with their functional issue first with one diet and protocols to clear up the problem, whether it's the fat digestion issues with the gallbladder or pancreas or absorption or carnitine shuttle or mitochondria, or they have a high carb issue and they need to clear up the infection. Once you've cleared those up, now you can switch your diet to your genetically ideal diet and go from there. So this is what I mean by people can find out their genetics, they can implement, but if they hit a plateau, that's when functional testing will come in. The problem is that people only do functional testing, but they never did their genetics. The diet they did to help them get off the functional issue will eventually backfire because genetically they're not optimized for that diet that was useful to get rid of the functional issue. So even if people just do functional, they need to do their genetics eventually because they're very likely to run into problems, and they're not going to see the weaknesses they've inherited in the future that are going to manifest most likely in the future. So I recommend everyone does their genetics and that they keep an open mind to doing functional testing down the line if they hit a plateau.
All right, makes sense. And then also, detoxification, which methylation is part of phase 2 detoxification, but you could also
Absolutely.
get a good amount of information from the genetics and see like if someone has the genes that cause problems with producing glutathione or
Absolutely. We'll even talk about that. I can pull up my presentation and show and actually discuss that directly.
Yeah, let's I mean, if you're ready to do that now, let's go for it.
Let me know if you see the screen.
Yep, it is up.
Okay, great. So what we're looking at here is just to go through the original reason we were talking about Gary Brecka's his 10x gene test, etc. And this is coming from his work. He tests for MTHFR, MTR, AHCY, which is the one I mentioned before, and COMT. And his claim was that these are the 5 most important things you need. Get these 5 done, and it'll just help you forever. And it's the most important genes for not only for methylation but in genetics, which I disagree with. And here's why. This is methylation, and this is not even showing all the genes associated with it. The genes are very complicated, and MTHFR is just over here, and that's actually an MTHFR2. There's a second one. There's MTHFR, the sequel, that is also involved there. And here's MTR, here's MTRR, here's COMT, and I actually had to add in the AHCY over here in the corner because it wasn't significant enough to even include in some of these genetic folate cycle, methylation cycles. So what you're looking at here, the whole point of this thing we're looking at here is how do you deal with homocysteine? Homocysteine is such a damaging molecule. The body has 3 separate ways to get rid of it. We have the direct pathway, which is the folate cycle, which is where MTHFR became most famous. We have the indirect pathway up through BHMT, which is to deal with it at kind of inefficient, and it burns through a lot of resources, but it's a way to quickly get rid of it. And then there's a way to turn homocysteine into glutathione. You have to go through the transsulfuration or the CBS pathway, and you actually have to go through some liver genes over here, including the a glutathione, a couple glutathione genes to actually synthesize glutathione. This is a phase 2 pathway. So when Gary asserts that it's MTHFR, MTR, COMT, and AHCY, it's like no, there's a lot more going on here, and additionally, there's other processes afoot. So I talked about inflammation as one of the 7 drivers. These are references to articles on PubMed that show how inflammation affects or controls methylation. Now, at a minimum, inflammation and methylation are bidirectional, so that they control each other back and forth. But there are very clear cases where inflammation actually controls methylation directly. It's not a two-way street. And these are the examples I mentioned, like the top 15 genes that control that are the most important 15 genes for inflammation. Well, here's a list of them. I mean, people can spend the time to go through the hexadecimal hieroglyphics here of all these different names and subnames of all of them. But basically, it's looking at the interleukin 1s, interleukin 6, interleukin 8, 18, TNF alpha, 3 CRP genes, 2 COX genes, and interleukin 10s. And a lot of gene tests don't even include the interleukin 10s, which is baffling to me, because the interleukin 10s are the genes that are responsible for putting out inflammation. I want to know, do I have a squirt gun or a fire engine on patrol to put out inflammation? Then we've got oxidative stress has its own control over methylation. So this is controlling free radical damage in the mitochondria. Again, more reference articles here. Here's a direct quote. Oxidative, just in bold here, oxidative stress on complex traits and disease may be partly mediated through changes in epigenetic markers, e.g. DNA methylation. So it's basically saying oxidative stress will change the entire methylation strategy of the body. So if you control free radical damage, then you change how methylation even works. Then we've got then we've got like the details of that. So for people who want to understand what dealing with free radicals actually means, is that the mitochondria is a very, very complex electricity factory, and it takes carbs, proteins, and fats, combines them with oxygen, and burns them to make electricity, or what we call ATP. Now, when you burn things in this factory, you make a lot of sparks, and those sparks are the free radicals. And so you need janitors to run around and put out the sparks so the factory doesn't get caught on fire. So the superoxide enzyme, MN-SOD, who I've kind of jokingly called Mr. SOD or MN-SOD, he's the head janitor. He's the head janitor. So the head janitor is Mr. SOD, and he takes 2 of these sparks and combines them together to form hydrogen peroxide. Then his 2 subordinate janitors, GPX1 and CAT, that stands for glutathione peroxidase and catalase, they take 2 hydrogen peroxide molecules and convert it into oxygen and water, which, if we pause to think about it, that's amazing. Your body will take free radicals and convert it into oxygen and water, which is incredible, absolutely incredible. So these janitors are really important. And if you have variations, like if you've got a red dot with Mr. SOD, like I do, then your head janitor is limping along and is not able to keep up very well. So I need to implement lifestyle and diet strategies to give Mr. SOD the support he needs to work efficiently to prevent the mitochondrial factory from catching on fire. Now, this relates back to, we can relate, your expertise in thyroid health. So inflammation, I'm sure you've had multiple podcasts on the role of inflammation, of how it affects thyroid. I mean, am I right? Like just tons and tons and tons, right? So wouldn't it be useful to know if inflammation is super important on thyroid health? Wouldn't it be important to know, like, if you are a hyper-inflamer like myself, are you more prone to inflammation than other people? So then you can implement additional strategies, genetic strategies, so that the other strategies that people are doing to improve their thyroid health work better. And that's the connection between genetics and thyroid health, or anything else. If you can help the inflammatory pathway, then all the other things, the specialized things for whatever organ du jour you're looking at, whether it's thyroid or adrenals or gut or whatever it is, then it's all going to work better. As it relates to mitochondria, what I learned about mitochondria, the best way I've heard it described is the mitochondria is the business end of the thyroid. That the mitochondria is the end organ of the thyroid. What is the target of T4 floating around? The way I describe it is T4 is this four-wheel car that drives around in the bloodstream, and then it pulls over into a cell right up next to the mitochondria factory, and it opens one door, and you hear the transformers noise, and it turns into T3 engineer, and it button mashes on the controls of the mitochondria to dial up the activity and the burn rate of the mitochondria. And the issue is that you can have great engineers button mashing on the control panel, but if the mitochondria is broken in some way, then the thyroid hormone, the engineers aren't there to fix the thyroid, they're there to dial it up and dial it down. So that's why mitochondrial health is critical for thyroid health. And so one of the links of genetics and thyroid health is that if your janitors inside the factory can't keep up with things catching on fire, then you're going to have problems implementing any successful thyroid strategy, because the end organ of the thyroid hormone is on fire because there's sparks flying everywhere. Does that resonate?
Yeah, it makes sense. Pretty much if you have like certain genetic variations and, inflammatory markers, or you mentioned like an oxidative stress markers, it's going to be really difficult to reduce inflammation, reduce oxidative stress. You're going to get mitochondrial dysfunction, which in
Absolutely.
turn can affect thyroid function. And then most people with thyroid conditions have thyroid autoimmunity, Graves, Hashimoto's, which associated with oxidative stress, inflammation. So yeah, I definitely understand.
Awesome. So we're gonna jog quickly we're gonna get to liver in a second. First, we're gonna talk about vitamin D utilization, and these were citations that vitamin D, that the combination has a role in regulation of gene expression, including DNA methylation by the vitamin D receptor. So what the big thing to understand about vitamin D is that it's been estimated between 3 to 5% of your entire human genome is controlled by vitamin D. That's 1/33 to 1/20th of all your DNA is influenced by this one molecule. I would put vitamin D as like one of the most important molecules in the entire human body as it relates to health. And if people are dealing with any type of, the main function of vitamin D is regulating inflammation and immunity. It's bone formation is like one small piece of it, but it's not even the major piece of the whole story of vitamin D. So people's abilities to utilize vitamin D is critical. And in fact, like this double red dot here, this is a pattern I've seen when running tests on hundreds and hundreds and hundreds of people, is that almost always when someone has a chronic issue or an autoimmune issue, they almost always have double red dots or double yellow dots in their vitamin VDR and VDR2 genes. And I noticed this as a pattern, and because I also found the exception proved the rule, is because I had one client who had really terrible long-term health of all sorts of stress, but had a double green dot. But that person went through a massive utterly life-changing, formative, long-term, stressful event and series of events. So the chronic breakdown was vastly influenced by an acute and chronic stress response. Everyone else who had struggled chronically with their health, I have seen a pattern of double red dots and double yellow dots in the vitamin D receptor genes. And I went back to the chief scientist and said, I want you to look at your primary database. I want you to talk to your other practitioners who do a lot of these gene tests. And I want you to check, am I making this up, or do they also see, and do you also see a correlation between chronic issues like autoimmunity and these vitamin D receptor genes? Months later, he came back to me. He said, you're exactly right. He says, they see the patterns. I see the patterns. This is something very significant, that it's the inability to use vitamin D has massive ramifications for long-term health. And it also, at a minimum, is bidirectional. It's in relationship with methylation, and also in times has control over methylation. So for anyone watching this or listening to this, I really, really encourage people to really get clear on their vitamin D receptor genes. And it's not just, the answer is not just taking vitamin D. I mean, that is helpful, but there's other things you have to do in order to increase the expression of the vitamin D receptors on the surface of the cell so it can capture more vitamin D. Simply taking more vitamin D may not be enough.
That was actually going to be my question. So taking massive amounts of vitamin D, like 10,000 IUs, obviously it's not a long-term solution, but then the question was, can you do anything about the gene since it's genetic, but you can change the expression, so it's not permanent.
Absolutely. That was the fourth criteria of what genes to pick. So you don't pick any fatalistic genes. Like, oh my God, you got this gene, you're done. Bye. You might as well write your will now. You may keel over tomorrow. Bye. And I call that 23andMe syndrome, where people get their genetics report and they freak out, and they're not empowered to actually know what they can do about it. And none of the genes I'm showing you here are fatalistic. That you actually can do things to support the expression the way you want. Then we mentioned like the detoxification. So there's 8 major genes in the liver detoxification, 3 in phase 1, 5 in phase 2. We already saw 3 of them up above in the methylation pathway here, these GCLC, GCLM, and GSTP1 genes in the formation of glutathione. So this is a very interesting diving into the effects of methylation and detox. And here's a couple quotes. Like, environmental toxicants such as toxic metals can alter epigenetic regulatory features such as DNA methylation. So this is right off the bat. It's saying that toxins can change how you methylate. So if you're not able to detox properly, then you're just going to change your methylation status. Then the other thing that Mr. Brecka talked about was how he said AHCY gene was the main gene that was really important for understanding people's mental moods. And I was like, there's other really important genes out there like MAOA. And the issue with the AHCY is that the main answer to it is B3, and there are very uncommon mutations of the AHCY gene to begin with. So I think that there's better genes to pick, and the ones that he did pick were not. The HCY was not. It's just B3 is all I could find. If he's got something else, I'd love to see the citation, but I couldn't find one. All I saw was like, it's B3, and away you go. And if there are other things, that's good to know. But it didn't really seem that practical. The MAOA genes are really interesting because there's very high correlations with different predispositions to different moods. Exercise and stress management helps a lot, but different combinations mean different things. This one's a public service announcement. The people who have MAOA1 red dot and MAOA2 green dot, alcohol is particularly dangerous for them because these are the people who can get like violently drunk. And this particular combination is really bad for people who drink alcohol. So this is a no-go zone for when I see this combination in the clients that I run their genetics for. And there's certain things that help, but there's also and one of those things, certain things that help is not just do something, but to avoid something. And alcohol is one of those things in this case. So, in summary, in terms of Gary's work, is that I think that it's way too simplistic. He just picks 5 genes for methylation when in fact there's about 12.5. And also methylation is subordinate to all these other processes like inflammation, free radical damage, vitamin D receptors, and liver detox. And there's other better genes looking for mood than AHCY. So those are my thoughts about this whole methylation fiasco that was kind of released into the zeitgeist in the late last year. And for people, I just want to pause there. Do you have any other comments or thoughts that you wanted to discuss around methylation?
Well, I mean, first of all, a great presentation. So thank you for sharing. Interesting. Like again, I think as practitioners, a lot of us, including myself, think about the impact of methylation on detoxification since it's part of, again, phase 2 detox. But then you mentioned that the toxins themselves can have a negative effect on methylation. So if you have heavy metals and other exposure to other environmental toxins, also that could be.
And also inflammation controls has subordinates,
Yeah.
is above detoxification. The best example I can think of is like if you're in your kitchen and there's a garbage can full of garbage and you have a priority to take out the garbage. Well, what if you walk into the kitchen and say the kitchen is, on fire? What's the priority? Putting out the fire or taking out the garbage? And this is what I've seen. I gave a lecture at a genetics conference called Exercise-Induced Obesity, which it sounds like a funny title, Exercise-Induced Obesity. And what happened is that I saw a pattern in certain clients where the more they exercised, the fatter they got. And it was this kind of inflammatory weight, like it was a water puffy weight. And what happened is they had a certain combination of genes where they were hyperinflammatory, plus they had genetic variations in their liver detox genes, particularly GSTP1, which was the rate limiting step for glutathione synthesis. And what happened is that they were over exercising. One person in particular, she joined a CrossFit gym and they're nuts. And she was over exercising like an hour a day every day. It was stupid. And their trainer just was being really dumb. And what happened was that overtraining became pro-inflammatory. And so her whole body became inflamed. And when your body becomes inflamed, you retain water to dilute the inflammatory chemicals to prevent them from damaging your cells and tissues. So you basically retain water to buy your liver and kidneys time to flush out the inflammatory chemicals. Well, she had problems in those. She was pro-inflammatory, particularly in the CRP genes, which the CRP genes are right here. They're the ones that control the acute phase inflammatory proteins in the liver. So if you've got, it was a perfect storm. If you create massive amounts of inflammation, you overproduce the acute phase inflammatory protein.
I think you froze, Dr. Sam, at least on my end. I don't know if it's me or you.
Oh yeah, I'm back.
Oh, you're back.
I had a small glitch. So if she overinflames and she has problems with over creating inflammatory molecules in the liver and problems with liver detox, what's going to happen is that your body's priority is now to get rid of all the inflammation, not to detox estrogen. So not only did she put on puffy weight, but her cycles went off. For the men who had this same genetic pattern, they didn't have a cycle to go off, but they all developed man boobs. It's because they had increased inflammatory weight, and they couldn't get rid of the estrogen that was building up. And so the estrogen stayed in their system and repositioned fat in their body onto their chest. So the answer for them was not, fewer calories and more exercise to drop weight. it was to put them on, drop the inflammatory input and stop over exercising, get them on an anti-inflammatory, liver detoxing, anti-estrogen diet. You remove all the exogenous estrogens from heated plastics and all the other, culprits of exogenous estrogen. And, her muscle tone came back, her cycles normalized. One gentleman lost over 40 pounds in 1 month, and the other gentleman lost 1 kg, which is about 2 pounds a week, for 14 weeks or so. And the man boobs went away, and the belly fat went away. It wasn't, they were overweight, but they were overweight genetically because they had they're pro-inflammatory and they couldn't detox properly. So I had to give them different protocols than the standard. You have to exercise harder and starve yourself, because the weight wasn't calorie based. The weight was inflammatory based. And and exercising was the trigger, not the solution to the weight. So this is where getting your genetics checked is super important, because people have different styles of weight gain. Some people are inflammatory water weight gainers. Some people are hormonal toxic weight gainers. And some people are fat calorie weight gainers, which is the ones we typically think about. But the most people I've ever seen, the vast, vast majority of people I've seen who have stubborn, puffy weight, they're not fat calorie weight gainers. They're inflammatory water weight gainers with a skosh of the hormonal toxic weight gainers.
So you mentioned the CRP gene. So if someone has, genetic variation, so they might be more likely to have inflammation, acute inflammation, thus having positive CRP. I mean, every now and then I'll come across someone who has, it's not uncommon to have like people with high CRP, but sometimes it's more challenging to decrease. And obviously sometimes it could be a challenge finding out the source of the inflammation, but could it also be partially that it's a genetic problem too? That just
it's
because
literally written right here. CRP is triggered by mainly interleukin 6 and associated with many chronic diseases. Orange and particularly red dot indicate increased production of CRP and increased risk of inflammation. So people who have variations can overproduce CRP. So the answer is absolutely yes, in my opinion, based on the genetic research.
And then one other question with this, since there are other genes associated with inflammation, just because someone has a negative CRP and let's say a negative ESR, that doesn't mean they're inflammation free.
No, because inflammation takes on many different forms, and there's multiple molecules associated with it. CRP, ESR is kind of like, it's triggered by so many different things. It's almost a non-helpful marker because it's so non-specific. Whereas there's other more specific inflammatory markers to look at, and some people are more genetically prone to create certain inflammatory processes over others.
Yeah, I wanted others to hear, because again, some people will get a CRP, they see it's negative, and once they understand that it's associated with inflammation, they just might assume, well, everything's good to go. I don't have an inflammation, but just wanted you to reinforce that there are other
Absolutely,
inflammatory markers.
There's many more things to look for than just CRP.
All right. Well,
I wanted to share if people were interested in getting genetics labs for themselves. So
I was about to ask you. I was just going to ask you, like, for those who are interested in learning more about this, like, what can they do?
So there's the 110 genes, approximately 110 genes that I run, they're broken up into six different panels that they're all are one thing. I'm not like picking out individual genes. We're do all of them, and they're broken up into 6 different parts. One is the Find Your Ideal Diet, which is the one I described before about the copy number, like the number of cannons. That's determined if you are more keto, more paleo, more Mediterranean, or more high carb, or some variation in between. And by Mediterranean, I don't mean the ingredients of the Mediterranean diet. I'm talking about the carb density of people in the Mediterranean. And there's plenty of other people around the world that don't have the foods of the Mediterranean diet, but they have a similar carb ratio panel. So that's what I mean. So figuring out what your carb tolerance is crucial. Trigger foods. There's some hardwired food triggers. Like some people are hardwired that they can't tolerate histamines, or they can't tolerate gluten because they're at risk of developing celiac disease. They can't break down the lactose enzyme. Some people are genetically, like, they can't tolerate alcohol. And some people, this may be very hard for some people to hear, some people can't tolerate caffeine. And there's a certain percentage of population, myself included, that get what's called, quote, caffeine-induced anxiety and depression. Caffeine-induced anxiety and depression. Now, when I talk to a room of entrepreneurs, and I mentioned that there's at least 10% of you that have this variation where you get caffeine-induced anxiety and depression, you can hear the stress response just go in the whole room, because everyone believes that their entire business runs on coffee. So that's a very confronting set of genes. I have those genes, and so I have found non-caffeine based ways to support my energy systems. And it doesn't matter how much coconut oil or MCT oil you put in your coffee, the caffeine will screw you up if you have these genetic variations. There's the vitamin D absorption genes, and that's like not only how you're able to utilize vitamin D to get it into your cells, but also the sunlight pathway. Like, there's about a half dozen genes between sunlight and your bloodstream in terms of synthesizing vitamin D. There's also eating behaviors. Some people are genetically wired to not feel full as readily when they eat food, because fullness is not in the stomach, it's in the brain. It's the signal from the stomach to the brain. Some people are genetically wired to crave more sugar, or to overeat sugar, or to avoid or not mind bitter foods. And all of these things really influence how you eat. And for some people are thinking, wait a minute, you just said there was no fatalistic genes you test for, but you said some people are prone to craving sugar. Isn't that fatalistic? It's like, no, it's just that if you have the hardwired genetics for that, then you don't try to control your willpower, you control your environment. So I don't keep bread in the house because it won't last half a day. So I'm not stronger than bread. I'm smarter than bread. So it's controlling my environment. So that's how you can be empowered by these seemingly fatalistic behavioral genes. It is not, don't bother with willpower, just control the environment. And there's very specific strategies with that. And then there's the immunity support genes, which is looking at immunity genes. In particular, there's lots of vitamin C and zinc genes. Some people just need more zinc and vitamin C. And then the achieve your natural weight genes. This is the big panel that has the genes that have those 7 drivers: inflammation, free radical scavenging, liver detoxification, vitamin D utilization, the dozen-ish plus methylation genes, the cardiovascular blood pressure genes, and the fat and energy metabolism genes. And that's in this one. This is the one where you look at what can I do to shore up those genetic fault lines, and what do I need to do from a genetic point of view to normalize and optimize my weight and decrease inflammation and all the rest of it. So that's how the 100 or so genes are broken down. This is just kind of more of a write-up of what all the different gene panels do. And then if people want to learn more, the next step is to go to my website, drsamshay.com, drsamshay.com. And there's several ebooks there on genes, and there'll be an invitation there to either join a webinar, or if you need to join a webinar or schedule a call to discuss the best option for you. And I also have on my website a Genetics Plus program, which is a way for people to learn their genes either in a DIY, do-it-yourself model, where you get the gene reports, the customized report, and then a back-end portal with lots of detail of how on all the major gene categories as a self-paced learning program. And there's also an office hours model if people want to have an option to be in kind of a group dialogue about genetic questions as they implement over at least people need to give themselves about 6 months to really implement the genetic changes. And once they implement them, you have them for life. Because again, these recommendations are based on the science. They're based on your genes, which don't change. And if people lock in these lifestyle changes, they have these benefits. They keep doing them. They'll have these benefits for life. And again, the investment in the genetics testing is the single highest value per dollar investment in health anyone can ever do, because the results do not change over time. The recommendations do not decay over time. You get this test once, and you've got the recommendations and the decision trees for life. And it's especially critical if you want to find out your ideal diet, what your genetic baseline is. So I very much encourage people to, at a minimum, get their genes tested.
All right. Well, thank you so much. I'll of course make sure to include the links, any links in the show notes. Obviously, the one to your website, drsamshay.com. And yeah, anything else that you wanted to cover or summarize before we wrap it up?
I'd say that uh, I'd say that the genetics is a very fraught ecosystem where everyone's got a very strong opinion, and each gene, if you talk about any individual gene, it's going to sound like the most important thing ever, like MTHFR, and it's going to come across as magic bulletism. Oh, if you just deal with this one gene, it'll just solve all of the things. That is simply not true. And this is why I recommend that if you're going to get genetics testing, make sure there is a priority filter put on the genes, and that you're looking at the highest priority genes as a whole, and that you have a logical, science-based readout of what are the things to implement in order of priority, and not fixate like a dog on a bone on one gene, or even just 5. Like really look at the highest level collection of all the genes, and really look at how you can best support your lifestyle over the long term. That the genetics testing will give you a roadmap long term. And also, if the gene tests, you hit a plateau, make sure that you open up your mind to, you may need to get some other things attended to in order to then further benefit from the genetic lifestyle. So genes themselves are not a magic bullet. They're not themselves a magic bullet. And it may seem strange for someone talking about genes for an hour to say genes aren't the magic bullet. They're not. They're a very, very, very critical, important part of someone's entire health plan, their entire well-being plan. Whatever your objectives are, your performance goals are, genes must be a part of it, but it is not the exclusive part of it.
All right. Well said. And just as important, also, I realized on that slide was a link to your stand-up comedy
Oh,
clip.
of course, yeah, yeah. The laughter
So
is the best medicine, of course. And
yeah, so I'll include those in the show notes too.
yeah, don't let the academic veneer fool you. Like, so what I'm doing is I specialize. My comedy is very clean, observational comedy, modeled after Seinfeld and Jim Gaffigan, and the story arcing of Seth Meyers. It's clean observational comedy that's very eternal, and the primary things I focus on are like the ridiculousness of people in the health industry, and whether consumers, like, whether you're in Boulder, Colorado, or Austin, or whatever. But I primarily focus on what it's like to have Aspergers and to be I don't like the term neurodivergent. I like to think of myself as neurodistinct or neurospicy. And I use stand-up comedy to educate the normies on what it's like to live in their world. And it gives language to the other aspies of like this. We have shared reality, and here's a way to explain it in a way that's palatable. So I use stand-up comedy to make the topic palatable, and then we use social media to make it scalable. And currently working on finish writing an hour special just on Asperger's, which hopefully will be first performed this upcoming summer.
All right. Well, wonderful. Yeah, that's yeah. And so you're doing that live? Like it's going to be a live presentation?
Oh yeah. So I'm waiting to hear back. At the time of this recording, I still have to hear back from the Denver Fringe Festival if I was accepted to have an hour slot in June. So I won't know until early March. And if I'm not accepted for whatever reason, it's fine. I'll find another venue, or I'll just have more time to work on it. So it's. I mean, I do. I mean, for people listening, like I do MC work, and I also do stand-up comedy at conferences that are relevant to the conference at hand. Like I was at Mindshare Community and I gave a 5-minute talk on how to put humor into your messaging for increased rapport, so that people could really resonate with the material and receive it better, and that there's actually a structure to incorporating comedy into your messaging. So I love comedy. I mean, that's a whole other topic we can talk about. And I'm sure we can talk about, just have a whole thing talking just about thyroid, and that's a whole other separate discussion. That's more on the functional side. That's going through all the tests around thyroid that a lot of people who are struggling with thyroid, they miss. So like thyroid and gut connection, thyroid adrenal connection, thyroid estrogen connection, thyroid and irons and other nutrient connection, thyroid and mitochondria connection, which we discussed, albeit briefly, today. And just knowing the entire constellation of thyroid tests, but the tests that surround the thyroid and connect to the thyroid, if people want to get a full view of how to optimally help their thyroid from a functional medicine lens.
Awesome. Yeah, might have to get you back for that. And well, thanks, Dr. Sam. Again, thank you for shared a lot of great information with genetics, including methylation. Definitely check out Dr. Sam's website, and also again the comedy clips, and again, it was just a pleasure chatting with you.
Same. Thank you so much for doing this. I really enjoy teaching, and I really love sharing this, and I'm so grateful to people like you who are doing the yeoman's work of educating the public at scale, and really helping empower people with great information from all the guests that you bring on.
Yeah, thank you so much.